Hello students. So today we are going to do the second part of cell structure and organization. In the first part we did the introduction of cell, the different microscope used to observe cell, the different types of cells, kinds of cell, that is prokaryotic and eukaryotic basic thing we learned, and we learned in detail about prokaryotic cell. Also about the totipotency. Now today we are going to learn about the eukaryotic cell. As the first slide you can see the eukaryotic cell, the nucleus in the center, mitochondria on the sides, lysosomes and the other components present in the cell, as we can use the term cytosol or cytoplasmic streaming. Right, now in this again today we are going to learn the first few parts or organelles of the eukaryotic cell, that is cell wall, plasma membrane, cytoplasm, endoplasmic reticulum, Golgi complex and lysosomes. The remaining part we learn in the third part of the lecture. Now moving on to the next slide, here we can see both the types of cells, prokaryotic and eukaryotic. Again as we move further, we should always have a comparison between prokaryotic cell and eukaryotic cell, which will help us to understand the difference as well as the different organelles present in both of them. So if you see prokaryotic cell, that is bacterial cell, the capsule, cell wall, cytoplasmic membrane, ribosomes, pila, flagella, the nucleoid, that is a simple nucleus, and the cytoplasm. Whereas in the right side eukaryotic cell, which is present in all the higher organisms, protists, fungi, animals and plants, and you can see various parts in it: cytosol, vesicle, centriole, mitochondria, microtubules, nucleus with chromatin, reticulum, that is nuclear envelope, nuclear pore, nucleolus, also Golgi complex, lysosome, flagellum, plasma membrane, smooth endoplasmic reticulum, ribosomes, rough endoplasmic reticulum, which we are going to learn in detail in today's part. Then just the comparison between the prokaryotic cell and the eukaryotic cell. As you already saw the image, again we can see the image as well as the differentiating or distinguishing points between both. If you see here, prokaryotic cell, no nucleus, or we use the term nucleoid. Here nucleus is present, no membrane enclosed in the organelles, whereas in eukaryote we can see all the membranes right from mitochondria, chloroplast, Golgi apparatus, etc. Single chromosome, which is why it is called as a nucleoid. Here chromosomes are in pairs, no streaming in cytoplasm or streaming in cytoplasm. Just now we saw in the first slide the cytosol or streaming movement. Then cell division without mitosis, whereas in case of eukaryotic, cell division takes place with the help of mitosis, that is equational division. Further, simple flagella. In case of eukaryotic, the flagella is complex one. Smaller ribosomes, here larger ribosomes, simple cytoskeleton, complex cytoskeleton, no cellulose in cell wall, here cellulose is present, no histone proteins, whereas DNA bound to histone proteins in eukaryotic cell. So these are the basic differentiating points between prokaryotic and eukaryotic cell. Further, now we learn the detailed part of eukaryotic cell. Cell in which the nucleus has definite nuclear membrane are known as eukaryotic cells. These cells exhibit presence of membrane-bound cell organelles. Example, cells of protists, plants, animals and fungi. The eukaryotic cells have different shape, size and physiology, but all the cells are typically composed of plasma membrane, cytoplasm and its organelles, mitochondria, endoplasmic reticulum, ribosomes, Golgi complex, etc. and a true nucleus with all the parts. Again I kept the image of a plant cell and animal cell, as eukaryotic cell has got all the components. So in plants and animals only the minor differences like presence of cell wall in plant cell, which we can see in the image, whereas in case of animal cell it is absent. Also the chloroplast is absent in plant cell and present in plant cell and absent in animal cell. So there are small minor differences. Now we go into the detailed part of the parts, that is components of eukaryotic cell. So first one, the cell wall. Cell wall, it is a rigid, supportive and protective outer covering of plasma membrane of plant cells, fungi and some protists. That is, it forms an outer covering, it is protective, supportive and it forms a hard structure or a rigid structure, and is present in all the plant cells and also in certain fungi and protists as well. Algae shows presence of cellulose, galactans, mannans and minerals like calcium carbonate in cell wall. In other plants, it is made up of hemicellulose, pectin, lipids and proteins. Microfibrils of plant cell wall shows the presence of cellulose, which is responsible for rigidity, means to maintain the shape or rigidity the microfibrils are present in the plant cells, and the outer, that is the cell wall, is made up of hemicellulose, pectin, lipids and protein component. Further, some of the deposition of cell walls are silica in case of grass stem, cutin in case of epidermal walls of land plants, suberin endodermal cells of root, wax, lignin. These are some other components. It gives shape to the cell and protects from mechanical injury and infections. In plant cells, cell wall shows middle lamellae, primary wall and secondary wall. Middle lamellae, it is thin and lies between two adjacent cells. It is the first structure formed from cell plate during cytokinesis. It is made up of pectin, calcium and magnesium pectate. Softening of ripe fruit is due to solubilization of pectin. So the fruit gets ripened because of this softening of pectin or the solubilization of pectin. Primary wall. In young plant cell, it is capable of growth. It is laid inside to middle lamellae. So primary wall is inner to middle lamellae. It is the only wall seen in meristematic tissue, mesophyll, pith, etc. Now we can see the secondary wall. It is present inner to primary wall. Once the growth of the primary wall stops, secondary wall is laid. At some places, thickening is absent, which leads to formation of pits, means it is not thickened, because of which there is formation of pits taking place. Then plasma desmata. Now what is this? Plasma desmata is cytoplasmic bridges between neighboring cells. So two cells are connected by this plasma desmata. Further, it shows pores between the cell wall and middle lamellae. So we can say the exchange between these cells also takes place and also forms a bridge between the neighboring cells. Further, cell membrane, plasma membrane or biomembrane, different terms, right? So as we heard about cell membrane, plasma membrane, it's also called as the biomembrane. Towards the right, you can see the structure of the cell membrane, where lipid bilayer, protein transport, phospholipids, proteins, carbohydrate chains are present. First point, it is thin, qualified structure present both extracellularly and intracellularly. Extracellularly, it is present around protoplast, and intracellularly, it is present around most of the cell organelles in eukaryotic cell. So in eukaryotic cell, it is present, whereas extracellular means outer part of the cell, outside of this cell. It separates cell organelles from cytosol. That is, the cell organelles which are present, it is separated by the presence of cytosol, as you saw in the first slide. Thickness of biomembrane is about 75 angstrom units. Under electron microscope, cell membrane appears trilaminar, made up of three layers. As the name says, tri, presence of three laminar or lamellae or three layers. It shows presence of lipids, mostly phospholipids, arranged in bilayer. Lipids possess one hydrophilic head and two hydrophobic non-polar tails. What is the meaning of this hydrophilic and hydrophobic? Hydrophilic, as the name suggests, it's electron loving, water loving, or here lipid possesses one hydrophilic polar head which has liking or absorbs water, whereas two hydrophobic which repels the water molecules. So phospholipids are amphipathic, means it is both. Amphi, as we have use the term amphibian, where it is present land and water. Similarly here, they are in phospholipids, they are both amphipathic. Lipid molecules are arranged in two layers in such a way that their tails are sandwiched between the heads, as you can see in the image part. So they come towards the central part. Due to this, tail never comes in direct contact with aqueous surrounding to the water or the fluid present in the surrounding medium. Cell membranes also show presence of proteins and carbohydrates. So cell membrane also has proteins and carbohydrates. Ratio of proteins and lipids varies in different cells. So it differs from cell to cell. For example, in human beings, RBC shows approximately 52% of protein and 40% of lipids. So more amount of protein present in RBCs and less amount of lipid. Moving further, to learn this, we come across the fluid mosaic model. It is most accepted model of cell membrane. It is proposed by Singer and Nicholson in the year 1972. According to this model, it is made up of phospholipid, bilayer and protein. So this structure which you saw previous slide, that is what is the put into fluid mosaic model, where it is made up of phospholipid, bilayer and proteins as well. So proteins can change their position. Some proteins are intrinsic, that is occur at different depths of bilayer, means present in the inner layer we can say. They span the entire thickness of membrane, so they are called as transmembrane protein. So when you say it is present or covers the entire thickness of the membrane, hence the term uses transmembrane protein. They form channels for passage of water. Extrinsic or peripheral proteins are found on two surfaces of the membrane. When you say extrinsic, the outer layer, or as the term says peripheral, towards the edges or the corner region. Qualified nature of lipid enables lateral movement of proteins. This ability to move within the membrane is measured as fluidity. Why fluidity? As it can move in the inner part. As again reminding the term or repeating the term, cytosol or cytoplasmic fluid membrane, where the components can move inside because of the fluidity. If it is rigid, hard, or if it is not fluid, then there will be no movement within the membrane, right? Okay, main function of plasma membrane is transport of molecules across it. This membrane is called as selectively permeable. So what do you mean by selectively permeable? It only allows certain molecules to pass through it, not all. Okay, during passive transport, many molecules move across the membrane without spending energy. Some solutes move by simple diffusion, means by directly diffusing into it without any much wastage of or utilization of energy, etc. Along the concentration gradient, it is from higher to lower concentration. Neutral solutes may move across the membrane by the process of simple diffusion. This is called as passive transport. So in case of the simple diffusion, the solutes can easily move through it. Further, water may also move by osmosis. So even it is by simple diffusion or even by osmosis. So during active transport, few ions or molecules are transported against concentration gradient from lower to higher. As we just learned, it is usually along the concentration gradient from higher concentration to the lower concentration. But sometimes it can be by active transport, which is vice versa, that is from lower concentration to higher concentration. It requires energy. For simple diffusion, energy may not be required, but in case of active form, energy is required. So ATP is utilized. As such, transport is an energy dependent process in which ATP is utilized. It is called active transport. Example, sodium potassium pump. That is, sodium and potassium ions are present. So hence this is one of the example. Polar molecules cannot pass through non-polar liquid bilayer, so they require carrier proteins. So when non-polar, that is molecules which cannot pass through the bilayer, non-polar layer, that's the polar molecules, then they require certain carrier proteins for moving across. Now we can see here the fluid mosaic model, which we saw in the previous slide. Extracellular fluid, protein channel, that is transport of protein, just now as we read, globular protein, glycoprotein, carbohydrate, hydrophilic heads, as we read, hydrophilic means water loving, phospholipid molecule, and bilayer hydrophobic tails, that is which repel or which does not take up water. For surface proteins, alpha helix protein, there is integral protein, globular protein, peripheral protein, glycolipid, cholesterol, all which is present in the cytoplasm. So this complete process was put up by Nicholson and Singer, hence termed as fluid mosaic model. Further, coming to the third part, cytoplasm. The cell organelles or cell contains ground substances called cytoplasmic matrix or cytosol, which I am repeating the term right from the first slide, which we saw how the movement takes place in the animation of the GIF. Even further, we can see some more animation related to it. This colloidal jelly like matrix is non-static. When you say static, you come across the term static electricity or because of movement of electrons, etc. So there is no static electricity or static movement. It shows streaming movements called cyclosis, that is as use the term cytoplasmic matrix or cytosol or cyclosis, the streaming movement of organelles taking place within the cell. The cytoplasm consists of water as major component along with organic and inorganic molecules like sugar, amino acids, vitamins, enzymes, nucleotides, minerals and waste products which are present. Hence, as we read in the previous slide in the image, as well as in the paragraph, the hydrophobic and hydrophilic. Since the water component is present, is a major component, the hydrophilic substances they readily take up water molecule. It also contains various cell organelles like endoplasmic reticulum, Golgi complex, mitochondria, plastid, nucleus, microbodies, and cytoskeletal elements like microtubules, that is in the cytoplasm, where in the further slides we may learn some of them, and the next part of the lecture the remaining all the organelles in detail. Okay, cytoplasm acts as a source of raw material as well as seed for various metabolic activities taking place in the cells. So cytoplasm has got all the source, or as we use the term raw material, which is required for carrying out all the activities or other metabolic activities. It helps in distribution and exchange of materials between various cells or cell organelles. Cell organelles are nothing but compartments in the cell that carry out specific function. So we can say all these cell organelles are compared as a compartment. So each compartment has a specific function or specific work to be carried out. Some of them coordinate with each other and complete specific tasks for the cell. As I mentioned, each one has a specific function to carry out. So nuclear membrane, endoplasmic reticulum, Golgi complex, lysosomes and various types of vesicles and vacuoles form such a group and together considered as endomembrane system. So now all this once I repeat these terms: nuclear membrane, endoplasmic reticulum, Golgi complex, lysosomes. What are these? The types of vesicles. Now all this movement of this vesicle we can see in the further I put animation slides where you can come across this movement of the vesicles, how it is carried out. Then further this is called as what? Endomembrane system. Further, organelles have distinct functions are not included in endomembrane system. Means one which has got particular function, distinct function, that all of you know. Mitochondria, one is the major ones, which is the powerhouse of the cell, and there is a chloroplast where the photosynthesis is carried out, specific type of energy conversion in the cells. Now here you can see cytoplasm, the image, entire image of inside this cell, where you can see first towards the right side the lysosome, cell membrane, cell lamina, the centriole, cytoplasm which is present, that is we can say the swimming pool where all the organelles are present in the pool: cytosol, Golgi apparatus, mitochondria. And towards the left corner end you can see the movement inside the cell, how it is taking place: cytoplasm and intracellular materials, or you can see the organelles and cytosol, but you cannot see the nucleus over there. That's why it's mentioned minus nucleus. Only you can see the organelles and the cytoplasmic movement. So intracellular material presence inside the cell, enclosed by a cell membrane, except the nucleus where you can see presently. Now coming to the next one, endoplasmic reticulum. Going to the first point, this little network within the cytosol is present in all the eukaryotic cell except ova and mature RBC. So eukaryotic cells, where endoplasmic reticulum is present in all the eukaryotic cell, but with exception ova and RBCs, mature RBCs. Under the electron microscope, it appears like a network of membranous tubules and sacs called cistern. So we can see numerous folds or sac-like appearance, cistern. So in the next slide, we can see the exact part of the endoplasmic reticulum. It forms more than 50% of total membrane of eukaryotic cell. So it forms a major part. This divides the cytoplasm into two parts, one with the lumen of the endoplasmic reticulum, that is laminal cytoplasm, and the other non-laminal cytoplasm, that is lies outside the endoplasmic reticulum. Membrane of endoplasmic reticulum is continuous with nuclear envelope at one end and extends till the cell membrane. So you can see this, usually the endoplasmic reticulum, they are attached from one end of the nuclear envelope. It thus acts as an intracellular supporting framework and helps in maintaining position of various cell organelles in the cytoplasm. What does this mean? So in that case, it helps to form a proper framework of the cell and also helps to maintain the position of the cell. The outer surface of endoplasmic reticulum membrane may or may not be studded with ribosomes. So you can see endoplasmic reticulum, certain part may be with a ribosome, certain part may be not. In that case, if it is present, then it is rough. If it is absent, then it is smooth ER, right? Smooth and rough ER differ in their function as well. Smooth ER is involved in various processes in different cells. Depending on the cell type, it helps in synthesis of lipids. Which one? The smooth endoplasmic reticulum. Like example, steroid secreting cells of cortical region of adrenal gland, testes and ovaries. Also detoxification of drugs and poisons, liver cells, and storage of calcium ions, and that is the muscle cells. Okay, now in case of rough endoplasmic reticulum, then what is the major function? Protein synthesis. Example, pancreatic cell that secrete insulin. Thus protein are secreted by ribosomes attached to rough endoplasmic reticulum and are called secretory proteins. These proteins get wrapped in membrane that buds off from the transition region of endoplasmic reticulum. Such membrane-bound proteins depart from ER as transport vesicles. So one which is separated from the endoplasmic reticulum, then those vesicles are transport vesicles, as I mentioned, which we can see very clearly in the next animation slide. Rough ER is also involved in formation of membrane of the cell. The endoplasmic membrane are grown or grows in place by addition of membrane protein and phospholipids to its own membrane. So it adds the membrane proteins and phospholipids, which already their own membrane is present. Okay, now here you can see what we read. Okay, the later point is portions of this expanded membrane are transferred to the other components of endomembrane. As we just read, it's all these vesicles are called what? Endomembrane system. Now first you see the left side. The image, the figure we can see endoplasmic reticulum, nuclear pore, nuclear envelope, where it is attached to the nucleus, right? Now you can see the endoplasmic reticulum towards the upper right end. the smooth endoplasmic reticulum. Lower part you can see cistern, that is the folds or sac-like appearance, cisternae. Rough endoplasmic reticulum as the ribosomes are attached onto it. Now come to the right side, upper part. Now you can see easily what we just know, the certain vesicles which separate out from the endoplasmic reticulum and they come out, and now that is what is called as the transport vesicle, which come towards the Golgi apparatus. And at the lower end also we can say the membrane system. Why here I am using the term membrane system? Because you can see the further upper region, which is the ER, the one which is on front, which is a Golgi apparatus, and in between the movement of the transport vehicles very clearly we can see coming towards the Golgi apparatus, and from Golgi apparatus we can see further that is exocytosis taking place, or we can see the transport vesicles moving out, etc., or the secondary vesicle or secretory vesicle we can say. That's why it is the complete endomembrane system we can observe. Now, next, the Golgi complex. Golgi complex or Golgi apparatus or Golgi body, different names. Various terms are used to denote this assembly, manufacturing, packaging and transport unit of cells. What is this? Manufacturing, packaging and transport unit. Okay. Golgi complex essentially consists of stacks of membranous sacs, which is again called as cistern, similar to that of ER. Diameter of cistern varies from 0.5 to 1 micrometer. A cell may have few to several cisterns depending on its function. So in the cell there may be 1, 2, 4 depending on the cell. The numbers may differ. The thickness and molecular composition of two cell membranes of a Golgi sac differ from one another. The Golgi sac shows specific orientation in the cell. Even their arrangement is also differs from cell to cell. Each cistern has forming or cis phase. Cis means on the same side, and maturing or trans phase, that is on the opposite side. So we can see two phases, cis and trans. Transport vesicle that pinch off from the transitional ER merge with the cis phase of the Golgi system and adds to its content into the lumen. Just now which you saw on the previous slide, how the transitional epithelial or endothelial reticulum which gets transported from the ER towards the Golgi body, and you can see that transport vesicle coming towards the Golgi apparatus, and then the contents are again added to the lumen of the Golgi body. So this explains why Golgi bodies are usually located near endoplasmic reticulum. Modified and condensed secretion leave Golgi through transphase again as membrane bound vesicle. Golgi body carries out two types of functions. So two functions, which are those? Modifications or secretions of ER and production of its own secretion. So it modifies whatever secretion is done by endoplasmic reticulum, and also it has its own secretion. So cistern contains specific enzymes for specific function. Redefining, or we can say refining of product takes place in orderly manner, systematic way you can say. For example, glycolipids and glycoproteins that are brought from ER lose certain sugars and regain others, thus forming a variety of products. So Golgi bodies also manufacture their own products. So we can say it is a factory or industry which also has its own production. So Golgi bodies in many plant cells produce what? Non-cellulose polysaccharide like pectin. Now you can see the images also. Before that, two more points over here. Manufactured or modified, all the products of Golgi complex leave cistern from transphase as transport vehicles, again which we saw in the earlier slide. While they are leaving from Golgi, certain markers may get impregnated on their wall so that they can identify the specific target cell or cell organ. So also when they move out from Golgi body, they have got their own marker cells or marker, so that it can be identified by specific target cell or where it has to exactly go. Now you can see two images. One, the picture of Golgi apparatus where you can see cistern, the folds, the cis face on the upper right end, northern end incoming transport vehicles, lumen, the lower end southern end transphase, secretory vesicles. Now this we saw in the previous slide where we saw the secretory vesicle moving out from Golgi body, whereas the transport vehicle coming towards the Golgi body. There is a cis phase and from transphase moving away. Now we can see the another image very clearly how the vesicles are moving. Right now, if you see observely the vesicles which are moving from Golgi endoplasmic reticulum towards the Golgi body, and if you observe carefully these vesicles, they got some markers in it, or we can say some particles present in it, which indicates what type of function to carry out, what has to be the target cell, etc. Okay, so here also you can see very beautifully the animation how the vesicles are moving from ER towards the Golgi apparatus. That is the reason why in all these cells the Golgi apparatus is always near to the endoplasmic reticulum. Okay. Now again a little bit of additional information. Also I have put another animation image over here. Firstly, always remember the cistern in Golgi body are not physically connected to each other as they are in endoplasmic reticulum. So you can see in ER they are interconnected. In Golgi body they are not interconnected. They are actually individual ones. According to recent studies, it is proposed that cistern of Golgi body themselves mature moving from cis to trans phase. It is called as cisternal maturation model. It is also said that some vesicle recycle their enzymes. They have been carried forward by moving cistern back to the less mature region. That is towards the right. You can see the nucleus, endoplasmic reticulum, and also you can see the proteins, the smooth endoplasmic reticulum, rough endoplasmic reticulum, the nuclear pore, and you can see how the transport vesicle which is moving from the endoplasmic reticulum towards the cis end of the Golgi apparatus, that is transport vesicle, and then from the trans end it is again removed out as the secretory vesicle, and then you can see cell membrane protein expelled out of the cell. Now the next one, that is lysosomes. Lysosomes can be considered as dismantling and restructuring units of the cell. You can know the term itself is understood. Dismantling, to dismantle, restructuring the units, that is to restructure, reshaping. These membranes bound vesicle contain hydrolytic enzyme. So here what is present? Hydrolytic enzyme. Now, the enzymes in lysosomes are used by most eukaryotic cell to digest or hydrolyze macromolecules. So what is the importance? Here it is very clear the function to digest the macromolecules. The lysosomal enzymes show optimal activity in acidic pH. Lysosomes arise from Golgi associated endoplasmic reticulum. So from where lysosome arise? From the Golgi body, again which is associated with the ER. The list of lysosomal enzyme includes all type of hydrolases. As we know, enzymes they end with E or ASCS. Right, so here amylases, proteases, lipases. Accordingly, what they digest. If it is protein, proteases, lipid, lipase, amylase or carbohydrate, amylases. These enzymes are in inactive state, means they are actually inactive earlier, are activated only when lysosomes comes in contact with one particular organelle to form a hybrid structure. After the action of enzyme is over, the lysosome is reformed and reused. So once the work is done, action is carried out, then again it is reused and reformed. Lysosomes are thus found in various structure forms and carry out various functions for the cell. Lysosomes are polymorphic in nature. That's why various structure and forms. Poly means many, morphic means shape. So they are found in different shape and structures. We can classify lysosomes as primary lysosomes, which are nothing but membrane bound vesicle in which enzymes are in inactive state. Again we can relate with the earlier Golgi complex or endoplasmic reticulum where vesicles keep moving from ER to Golgi and from Golgi vesicles to further. Secondary lysosomes or hybrid ribosome, which formed by fusion of lysosome with endocytic vesicle. containing materials to be digested, represented as heterophagic vesicle. This is larger in size than primary lysosome. Residual body is the vesicle containing undigested remains left over in the heterophagic vesicle after releasing the products of digestion in the cytosol. So all the products after digestion, it is released into the cytosol. Lysosomes which bring about digestion of cells own organic material like damaged cell organelle are called autophage vesicle, or we use the term suicidal bags. So that is why the lysosomes are also called as suicidal bags. Further, an autophage vesicle essentially consists of lysosome fused with membrane-bound organelle or organic molecule to be recycled. Remember, every week a human liver cell recycles half of its macromolecule. So in liver cell, the half the cell is recycled. Lysosomes bring about intracellular and extracellular digestion, that is inside the cell as well as outside the cell. The intracellular digestion is brought about by autophagic vesicle or secondary lysosome, which contains foreign materials brought in by process like phagocytosis, example food vacuole in amoeba, or macrophages in human blood, that engulf and destroy harmful microbes that enter the body. Extracellular digestion is brought about by release of lysosomal enzymes outside the cell. Example, acrosome, a cap-like structure in human sperm, is a modified lysosome which contains various enzymes like hyaluronidase. So remember the enzyme hyaluronidase, which is present in human sperm, which helps to enter inside the ovum. These enzymes bring about fertilization by dissolving protective layers of ovum. During metamorphosis process found in many organisms, lysosome enzymes help in reusing the tissues of redundant organs. They also help in destruction of malignant cells, example T lymphocytes. Further, lysosome always remember, lysosome enzymes do not digest their own membrane proteins. Three-dimensional shape of these proteins probably protects the membrane. Accidental release of lysosomal enzymes in limited amount does not harm the cell because pH of cytosol is near neutral. Any insufficiency in secretion of lysosomal enzymes lead to disorder, which is that genetic disorder. That's why genetic disorder also takes place because of the insufficiency or sufficiency of secretion of lysosomes, such as Tay-Sachs disease. Due to insufficiency of lipase, brain gets impaired due to accumulation of fats. Now in the previous slide there was an image of showing the lysosome. Here we can see the animation image very clearly. Towards the right corner end we can see the purple colored nucleus part, and then the endoplasmic reticulum, and then the Golgi apparatus, and then you can see the vesicles which is moving towards the lysosome, or we can see the digestion which takes intracellular digestion, we can say. Now we read about Tay-Sachs disease due to insufficiency of secretion of lysosome. So here I just put an image for an understanding what is that exactly. A genetic metabolic disorder caused by deficiency enzyme hexosaminidase hexa that results in failure to process a lipid called GM2 ganglioside that accumulates in the brain and other tissues, which you can see how the accumulation taking place in the brain. Then Tay-Sachs disease is a rare inherited disorder. It causes too much of fatty substance to build up in the tissue and nerve cells of the brain. Then on the right side you can see brain damage, cataract, jaundice, enlarged liver, kidney damage. If a galactosemic infant is given milk, unmetabolized milk sugar builds up and damages the liver, eyes, kidney and brain. So see the effect of the accumulation or insufficiency of secretion by the lysosome. So that's a little bit detail about the Tay-Sachs disease. So that ends with our second part of the topic. Here it is a assignment question based on the today's part of cell structure and organization. The third part, in which we'll be learning about mitochondria, the nucleus, chloroplast, etc. So thank you all of you.